Hello everyone, I'm Kerwin Chen, a rising junior from Cornell University majoring in statistics. Today I'll be presenting my project on automatically mining and serving gene expression signatures from the Gene Expression Omnibus for gene-centric reverse search. A bit of background first, RNA-seq technology has enabled accurate profiling of genome-wide mRNA gene expression. This raw RNA-seq data is publicly available on the Gene Expression Omnibus to allow researchers to reuse and perform retrospective analysis for potential drug repurposing efforts or to elucidate new biological findings. However, the data is non-uniformly processed and generally provided in raw sequence form. Data harmonization efforts by Dr. Alex Lockman in our lab has resulted in the ARTIS-4 database, which contains more than 750,000 uniformly processed RNA-seq data. For this project, the ARTIS-4 data was used to automatically generate and serve gene expression signatures for human and mouse. The data processing workflow started with the human and mouse RNA-seq data from ARTIS-4, which was then filtered for bulk RNA-seq data, metadata elements identified, the samples labeled against a dictionary of control and perturbation terms, and a list of GSMs generated for signature calculation using lemma. A bit more about the labeling and scoring mechanism. To label the samples, frequent terms for control and perturbation were first manually labeled, and then the set of words in the sample label were compared against this list of frequent terms to determine a control, perturbation, or undetermined label for that sample. The labeled sample groups were then grouped into three categories: undetermined, control, or perturbation, and then studies were grouped based on this ordered triple. Signatures were then given a confidence score based on a penalty system of summing up the number of extrapolations needed, for example, labeling and direction of expression to calculate a signature. The score ranges from 0 to 3 inclusive, with 0 being the highest confidence. To give a concrete example of the whole signature generation pipeline, we looked at this particular study on the regulatory effects of HNRNPL and LARP on RNA expression in LNCAP prostate cancer cells. On the left, you can see a screenshot of the study on the GEO page with the title circled in red. The page also provides additional information regarding the experiment type, summary, overall design, contributors, etc. On the right, you can see a screenshot of the same study on ARTIS-4 with the uniformly processed samples available for use with their sample GSM IDs, title, organism, and library size. The samples are taken from ARTIS-4 and grouped and labeled as control, perturbation, or undetermined. We see here that three groups were found: the SI-HNRNPL replicates, the SI-CTRL replicates, and the SI-LARP replicates, and one of them, which is highlighted green, was identified as the control group due to the keyword found in the sample titles. The other two groups are undetermined, giving the order triple 210. Each of the undetermined groups were then paired with the control group to calculate a signature each, for a total of 2 signatures. Since the undetermined groups were extrapolated to be perturbations when calculating signatures, that's a +1 penalty, giving each signature a final score of 1. A signature with a score of 0 would denote the best signatures with no extrapolations needed. So in other words, when all samples were successfully labeled as either control or perturbation. So these are some statistics regarding the calculated signature data with human on the left and mouse on the right. For human, there were almost 3,000 studies, which corresponded to about 4,000 signatures. About 72% of the human signatures were given a score of 0 or 1. For mouse, over 3,000 studies corresponded to about 4,000 signatures, with 76% of them given a score of 0 or 1. The score breakdown for human and mouse is also tabulated and graphed as shown here. The signatures are served on the gene-centric GEO reverse search apparatus for user query, and the structure and design of the apparatus was based on Sherry's apparatus project on type 2 diabetes from our lab. On the left, you can see the input page where users can select the species and input a gene of interest into the autofill box. Upon submitting, users can see a volcano plot of differential expression of the given gene in signatures of the species of interest. Traditionally, a volcano plot would show all the up and down regulated genes for a single signature, but our lab has repurposed the volcano plot such that a plot is instead generated from a single gene, and each point represents a signature from a study in which the gene is over or under expressed. In this example, the differential expression of AKT1 across all the calculated human signatures is shown. Hovering over each point also allows you to see information on that signature, including the name of the study it came from, the score, p-value, adjusted p-value, and fold change. The results are also tabulated for signatures where the gene is up or down regulated. The table contains the information found when hovering over the points in the volcano plot, as well as a link to the original GEO study. Full results from these tables are also available for download as CSV files. Additionally, if the gene is orthologous between human and mouse, then the corresponding information will be shown for the other species as well. As a case study to understand the utility of the appeter, we looked at SARS-CoV-2. It has been established that ACE2 is a cellular receptor for entry for the SARS-CoV-2 virus, and a recent review suggests that patients with overexpressed ACE2 are more susceptible to COVID-19 infection, while patients with underexpressed ACE2 are more susceptible to COVID-19 complications due to the important homeostatic role that ACE2 serves in the renin-angiotensin-aldosterone system. Therefore, it's important to identify drugs or diseases that modulate the expression of ACE2. Using the appeter to query ACE2 in humans, various signatures were identified that modulate ACE2 expression. A549 cells infected with the influenza virus were shown to underexpress ACE2, potentially suggesting an increased susceptibility to COVID-19 complications for patients dually infected with SARS-CoV-2 and flu. Looking at ACE2 overexpression, lung-derived cells exposed to e-cigarette vapors were shown to upregulate ACE2 expression, potentially suggesting an increased risk of COVID-19 infection for e-cigarette users. Recent literature from researchers at Stanford has shown an association between vaping or using e-cigarettes and COVID-19, in that diagnosis was more likely among e-cigarette users. Additionally, nasal epithelial cells stimulated with the SLR14 ligand and human-induced pluripotent stem cell-derived cardiomyocytes treated with Melphalan, a chemotherapy drug, were shown to upregulate ACE2 expression as well. These molecules can therefore be further researched as potential treatments to alleviate COVID-19 complications. In fact, low-dose inhalation of Melphalan is currently tested as a therapeutic for COVID-19 in clinical trials. In conclusion, the appeter serves as a hypothesis generation tool for retrospective analysis of RNA-seq data for drug repurposing efforts and many other applications, as demonstrated through the COVID-19 case study, where drugs and diseases that could be further explored as risk factors or treatments for COVID-19 were identified. Another interesting case study would be to identify drugs or diseases relating to aging and its associated gene clotho, but this is not elaborated on for the sake of time. One final note is that the top, up, and down regulated genes from the signatures are also processed into gene set libraries by Max and Sherry from our lab for reference in Enricher. In regards to future steps to improve the appeter, that would include using machine learning models to better identify control and perturbation samples. Furthermore, more work could be done on extracting key metadata elements from samples and studies and mining additional signatures from ARTIST-4, including from studies with 2 or more control groups, as well as from studies with more complex experimental design. Lastly, I want to thank everyone in my lab, especially Dr. Alex Lockman and Dr. Mine for their support of the project, Megan Wajalowitz for helping with the signature calculations, Sari Shea and Daniel Clark for the code and appeter support, and the rest of the summer scholars. I also want to give special thanks to Sherry Jenkins, who organized the program.